Today we have the pleasure of hosting Dr. Judy Cho, Dr. Stuart Scott, and Dr. Owusu Obeng for a talk on the Charles Bronfman Institute for Personalized Medicine. Dr. Judy Cho is the director for the Institute of Personalized Medicine and the Ward-Coleman Professor of Translational Genetics and Medicine at the Acting School of Medicine at Mount Sinai. She is the principal investigator and chair of steering committee of the NIDDK IBD Genetics Consortium. Her laboratory is presently investigating the role of less common variation in innate and epithelial cell immunity, contributing to IBD in the Ashkenazi Jewish population. Dr. Stuart Scott is an assistant professor in the Department of Genetics and Genomic Sciences and is an American Board of Medical Genetics and Genomics certified associate director of Mount Sinai Genetic Testing Laboratory. In addition to clinical laboratory diagnostics, his research interests have focused on translational molecular biology, particularly in the areas of pharmacogenetics, cytogenomics, and epigenomics. Additionally, he is a member of the Clinical Pharmacogenetics Implementation Consortium and has written practice guidelines for pharmacogenetic guided therapy. He's a member of several international pharmacogenomic research consortia, as well as a co-investigator of the Mount Sinai Institute for Personalized Medicine pharmacogenomics program that is implementing preemptive clinical pharmacogenetic testing. Dr. Owusu Obeng obtained her Doctor of Pharmacy degree from the Albany College of Pharmacy and Health Sciences in Albany, New York. Currently, Dr. Obeng is an assistant professor at the Acting School of Medicine at Mount Sinai in the Institute for Personalized Medicine, where she's leading efforts to translate pharmacogenomics applications into clinical care. Her research interests lie in developing processes, best practices for clinical implementation of pharmacogenomics. Moreover, Dr. Obeng is the clinical pharmacogenomics coordinator in the pharmacy department at the Mount Sinai Hospital. Please join me in welcoming Dr. Judy Cho, Dr. Stuart Scott, and Dr. Owusu Obeng.
Thanks so much for that kind introduction. So the introduction to the Charles Bronfman Institute for Personalized Medicine, the word introduction has a twofold meaning. I'm relatively new to this position, so I'm still being introduced to the IPM. Started this in November of 2015. And then for those of you who are less familiar with the IPM, I think this may prove to be a useful introduction to the IPM. So we want to divide this talk, this whole hour, in about 3 different parts equally. So I'll provide an overview of what precision medicine is, talk about precision medicine vis-a-vis GWAS, and then talk about the administrative structure and provide you a broad overview of the structure of IPM. The second part will be a tag team between Stuart Scott and Aniwaa talking about pharmacogenetics and pharmacogenomics, both discovery and implementation. And then I'd like to leave plenty of time for a panel discussion of the above group, as well as Steve Ellis, who's the director of computer IT and IPM, as well as Amanda Merkelson, the managing director for the Biobank. So precision medicine, it's a hot term these days, and it really started for the past few years, but it really was catalyzed in the State of the Union address from last year from President Barack Obama at the time, announcing at the time in a very tight and ambitious timeline, launching this Precision Medicine Initiative, or PMI, for a very ambitious part that is closely partnered with the Affordable Care Act. And so I have to admit, I didn't listen to the State of the Union last year. And so my first introduction to the whole idea of a PMI initiative came from the seminar series that I've hosted with Eric Schatt and Pamela Sklar called Genetics, Big Data, and Medicine. Our kickoff speaker, quite fortuitously, was the head of NHGR, Eric Green, and he's the one who basically introduced this very ambitious PMI program, who's the head of NHGRI, and you can, if you just go on YouTube and then YouTube Precision Medicine and Eric Green and Mount Sinai, you'll get this really nice seminar that Eric Green did outlining this. And so this is a lot of collaborative work. I was asked a few years ago to give a talk on big data and collaborative data and how you do this. And so I was worried that the talk was coming off as too abstract, and so I had a series of Warren Buffett quotes interspersed throughout, which kind of provided a skeleton for the talk. And then afterwards, I think people were puzzled. They go, why did you have so many Warren Buffett quotes throughout? And I didn't really nail it. I thought, well, who doesn't like Warren Buffett? But basically, the reason for this is that fundamentally why it makes sense in these big initiatives such as IPM or PMI is they represent investments on the part of the NIH, investments on the part of the medical school to make more profound progress in biomedical research and human health. So the credit for IPM really needs to go first with Erwin Bottinger. Erwin had a vision more than 10 years ago of personalizing medicine specific to individuals. And so he was from the Department of Medicine, Division of Nephrology, and founded the Bronfman Institute for Personalized Medicine, or Precision Medicine, their equivalent, in 2007. And so that beginning corresponded somewhat with the advent of genome-wide association studies, and the course of these two events largely overlap each other, and they're often conflated with each other, but they're quite distinct. And so a lot of the immediate gain from the BioMe and the Biobank has been through GWAS, but really I think its greatest promise is moving forward, because I think it was truly visionary on the part of Erwin to really have a very forward-looking structure for BioMe and the IPM, which we'll talk about in the next few minutes. So back to GWAS. So GWAS started in earnest about 2006, and this was a paper that we published with the NIDDK IBD Genetics Consortium in Science in 2006, and it was a big hit, and it was very exciting at the time. We identified interleukin-23R as a big hit that was common to both Crohn's disease and ulcerative colitis. So IBD affects about 1.4 million Americans. It's equally divided between Crohn's disease and ulcerative colitis. We had discovered NOD2 several years before, and NOD2 is specific to Crohn's disease, whereas IL-23R was one of the first IBD generic association signals. The association signals were, there were multiple in the region, and of these multiple independent alleles in the IL-23R region, the one that was most statistically significant and most interesting functionally was for a polymorphism at codon 381. So the wild-type allele is arginine. The less common or derived allele is glutamine at codon 381. And if you're one of those 1 out of every 7 European ancestry individuals who are heterozygous carriers for the glutamine allele, you're protected against developing IBD 2 to 3-fold. Okay? And so far as we can tell, these individuals that are glutamine carriers are completely normal, healthy, and happy. They have no, to what we can tell, increased risk for infectious diseases. And work from multiple laboratories following close on the heels of the initial GWAS discovery was that if you're one of those glutamine carriers, it's a relative loss of function. So if you carry the glutamine allele, you're less likely to develop, not only are you less likely to develop IBD, but in terms of the efficiency of IL-23 pathway signaling, the glutamine carriers are a relative loss of function relative to the arginine major allele. So you put those two facts together, points 2 and points 3 together. If you're protected against developing disease for carrying an allele, which is a loss of function allele, that would then argue, perhaps, that blocking the IL-23 pathway could then provide therapeutic benefit, which subsequently has proved to be true. So blocking IL-23 through anti-P40 antibodies are already approved for the treatment of psoriasis. Phase 2 studies have been published in IBD in New England Journal and are positive, and phase 3 studies are looking very positive. So I think it's just a matter of time before blocking P40 is approved for the treatment of inflammatory bowel disease. The genetics informs the therapies, and we know this. One of the earliest finds from GWAS is that a lot of the major GWAS signals that we saw across diseases went across the major autoimmune or chronic inflammatory diseases. And so this is comparing in the first column a variety of different autoimmune diseases. And pay attention to the next 2 columns. It's the MHC region, which across autoimmunity is by far the most important GWAS signal. And you can roughly divide the MHC signals into being class 2, where CD4 T cells are what's driving disease, or class 1, where it's the CD8 T cells that are driving disease. And importantly, even though you see that IL-23R was the big signal in the IBD GWAS, it clusters around certain diseases. It's IBD, psoriasis, interesting psoriatic arthritis, as well as ankylosing spondylitis, is where in European ancestry populations you see this association of IL-23R. Importantly, there's a whole cluster of different chronic inflammatory diseases for which you don't see any evidence of that same IL-23R signature, most notably rheumatoid arthritis. And again, this has not been published yet. I saw this in a press release over the summer, is that targeting that anti-P40 antibody that targets both IL-12 and 23 has actually failed in phase 3 studies in rheumatoid arthritis. So even though it worked in psoriatic arthritis, again, look back. So if you look at psoriatic arthritis, psoriasis plus joints, you have some IL-23R signature, it works, blocking P40 works in those cases of psoriatic arthritis, but it doesn't work in rheumatoid arthritis. So this is a specific example of where the genetics informs the therapy, and you can save potentially millions of dollars. And so what I'm interested in doing is expanding this concept focused on the Ashkenazi Jewish populations, where we can actually scale this. And there's, again, we don't have time to go into the reasons why the Jewish population may be particularly valuable. But if we do large studies of many diseases in Ashkenazi Jewish populations and try to identify similar alleles, relatively uncommon, protective, loss of function alleles, that provides a capacity to identify specific therapeutic targets across many diseases. And so that's a new initiative that we're about to hopefully undertake soon. And not only does it provide very specific targeted insight into, okay, which particular therapeutic target do we want to highlight, it also provides critical safety information as well as efficacy. Because if you have a situation where individuals are walking around with a loss of function allele, you study a lot of those individuals that have that loss of function allele, and there's no evidence that there's any deleterious effect from carrying that loss of function allele, what that tells you, it's not only going to be efficacious, it'll be safe in treatment of these disorders. So I think that this is a very effective way that we can potentially identify many few therapeutic targets quickly. So to summarize GWAS in 2 slides, 10 years of GWAS, so the good. And so in the case of IBD, we started touting the number of loci a lot. And so you'd see in the GWAS studies that would be published in Nature Genetics, okay, we're up to 38, we're up to 72, 163, and now we're over 200 loci. So that's the good news. And so each one of these definitive loci, they're definitive because you have p-values of less than 10 to the minus 8th. That's not going to occur by chance. These are true findings. They provide many new novel gene targets and potentially many new insights into disease pathophysiology. I've showed you for the second bullet point the value if you compare and contrast against chronic inflammatory diseases. So for many years, what are the new IBD targets? It was usually whatever was the rheumatoid arthritis target. Okay, well, so perhaps we have some data using GWAS whereby we can do this more effectively than simply just copying the rheumatologist. And in fact, that's not going to work in the case of anti-P40. So the comparative differences in the GWAS signals across different diseases may inform therapeutic prioritization. And the other kind of major thing, there's a seminal paper that I refer people to by PharmD et al., published in Nature earlier in 2015, which looked across autoimmune diseases. And they identified that 90% of the GWAS signals are non-coding in nature. And so they probably subtly affect gene expression in some way. And these causal, if you can call that non-coding alleles are going to be clustered within non-coding regions defined by DNase hypersensitivity, ATAC, seq, and so forth. And really what was interesting in that PharmD et al. Nature paper is that you can cluster and see enrichment of these GWAS signals in cell-specific enhancers defined by ChIP-seq and H3K27 acetylation or H4K4ME1. And that those cell-specific enhancers, if you compare, is the enrichment greater in T cells or B cells or monocytes or epithelial cells, those cell-specific enhancers will provide specific insights into mechanisms of pathophysiology for these chronic inflammatory diseases. So that's the good news. The bad news is that within these many loci that you can identify for chronic diseases, the causal gene may not be necessarily easily identifiable, and it may be impossible in many cases. So typically, if you take a locus, you use some arbitrary distance of, like, so let's look at a 1 megabase window around the maximal association signal. The causal genes may not be identifiable. The common variants are of extremely modest effects. They have odds ratios that are clustering around 1, so despite very large significance estimates, p-values, the significance is a function of the effect size as well as the sample size. These GWASs are massive. They're including like hundreds of thousands of cases. And so you can identify definitive signals of very modest effects. And so they're not actionable and they're not predictive. And so where we are in the genetics of complex diseases are looking at rare variants. So people are now undertaking these massive sequencing efforts where we're trying to find these rare variants that are kind of 1% allele frequency in a population and lower. You're going to see important population differences between them. And we've learned a lot about doing good clinical and translational research. You need to do large data sets. You need to QC them very carefully. You need to control for subtle population differences between individuals, and you can establish standards. And so this discipline in clinical and translational research has been valuable. But the limitations of this is that these are kind of advances of the same kind. It's basically fundamentally limited by the case control paradigm which I would argue is fundamentally limited. And the dimension that the case-controlled GWAS paradigm lacks is time. So going back to Warren Buffett, always invest for the long term, kind of a more homily, someone sitting in the shade because someone planted a tree a long time ago. His insights into the principles of human gestation. And then another point to this is that this is from Nate Silver's book of why weather forecasts are most valuable in the near future. And this has to do with Bayesian analysis. Don't ask me about the equations of Bayesian analysis, but you can only predict things shorter in the future compared to long term. And so if you talk about GWAS, you're looking at germline genetic variation at the very beginning of things, at the beginning of life. And so really where the field needs to go, because there's a very hard ceiling to the GWAS paradigm, and we need to break out of this. And I think this is, again, where the IPM and precision medicine initiatives, I think, play a key role. So in the field of autoimmunity, the value is that really what you look at is disease. For most of these chronic diseases, they occur over a lifetime. You have these early exposures. Epigenetics, the human microbiome, will shape the nature of the germline architecture you got from your parents. In the case of IBD, well before the patient presents with disease, you're going to have cases of altered immune function, which is measurable for many years prior to disease presentation, and then in autoimmunity and IBD, what you're trying to do is control structural damage. And so if you look at the therapeutic paradigms going in IBD or other chronic inflammatory diseases, when I first started as a GI fellow in 1991, IBD was treated usually by symptom-based treatment, so control the symptoms. The standard of care now is to control inflammation and prevent structural damage. We're going from mucosal healing in IBD. And the future then is going to be earlier intervention, tolerance reduction, and normalization of immune function. So that's past, present, and future. So what's precision medicine? There's a nice review from Collins and Varmus on this. Prevention and treatment strategies that take individual variability and their life history into account. There's a number of reasons why it's hot now. There's the need for the acceleration of clinically relevant advances in data, molecular, and social media that causes this. And they've announced, again, we're hopefully going to submit in the next day or two, our application to be part of the National PMI Initiative, a 1 million person cohort that's going to involve a lot of pilot studies, regulatory reform, active patients, and really the principle, one of the principles of PMI is that you're going to place the patient firmly at the center of this research and clinical care team. So the challenges of precision medicine, you're going to have to bring together people who speak different languages. There are multidisciplinary languages, and again, never underestimate that. You kind of understand this idea, but never underestimate the importance of jargon and how people think about problems in impairing communication between them. People exist in silos. There's a natural desire to collect everything. And again, as Warren Buffett said, diversification is a protection against ignorance. It makes very little sense for those who know what they're doing. So again, if we don't have a structure for the study, there's this natural tendency to want to collect everything, but that's going to slow things down if you really want to do a big, well-powered studies. And you do need big powered studies, because if the goal of precision medicine is to take all cases of Crohn's disease and divide them into pathophysiologic subsets, you're going to need massive numbers in order to do this well. And it's going to be costly. So when I took over the IPM directorship in November, the charge from Dean Charney was easy, right? No, transformational discoveries to benefit patients and to break down silos. And why is it an institute? So one type of silos is we silo ourselves along departments. You have the Department of Medicine, the Department of Surgeries, and the purpose of the institutes is to cut across those departments. And so what are the sources of silos? Tribalism. Again, you can think of that tribalism across subspecialties, across disciplines. Geneticists think differently than epidemiologists, who think differently than molecular biologists. There are habits, there are financial reasons for the silos. And again, not all silos are necessarily bad. We only have limited time and attention, and there is a need to focus. And so really the goal over the next few slides is to identify strategic and efficient collaboration. So the structure of IPM, this is the administrative team. There's an administrative component. There's a recruiting component, a core laboratory, which very efficiently can sample these large number of samples. You'll hear about the computer informatics from Steve Ellis. The Biobank is managed by Amanda Merkelson. And then I'll show in the next few slides some introduction to the IPM faculty. So in terms of the Biobank, it's the best in the city. There's basically over 33,000 patients that have been prospectively consented for prospective EHR mining and the capacity to recontact them. Now, in reality, it's not that easy to recontact, but it's remarkable. The regulatory care that the IPM team took in order to be able to mine this biobank prospectively is valuable. Uses just a single tube of blood, over 650,000 samples tracked. You get these very useful ERAP dashboards which can track recruitment and core laboratory function. Computer IT, this involves many complex components. It's the recruiting part, which is ERAP, the laboratory management part, the clip merge part, which we'll talk about more in the pharmacogenetics part. This has really been powered by Mount Sinai has a wonderful clinical data warehouse, which brings together all of the different clinical databases, as well as scientific computing and Minerva. So it really very, very sophisticated computer IT at IPM, powered by the larger clinical infrastructure. Just a few slides on introducing the faculty. So I was very fortunate that Ruth Loose agreed to serve as co-director of the IPM. You had that email alert about the most cited investigators in 2015, and Ruth made this. There are some diseases that are more important than IBD, I grant you. Obesity and cardiovascular disease might be some of those. So Ruth is an epidemiologist, a geneticist, genetic epidemiologist. She's interested in some counterintuitive ideas. What's the value or what's the nature of healthy obese people or unhealthy lean people? She's interested in designing nested studies within the larger BioMe cohort. Collaborations, again, feel free to reach out to her. She's extraordinarily collaborative. She has a ton of publications. Anyone interested in exploring BioMe data, physiologists, biologists, and she's very interested in this cohort of naturally lean individuals. Emer Kenny, so Ruth is in the Department of Preventive Medicine. Emer Kenny is in the Department of Genomic and Genetic Science. Her interest is looking at population differences, transethnic studies. She's really interested in Hispanic populations. She's very effectively mined the BioMe Biobank to look at monogenic forms of complex diseases and conversely, common forms of Mendelian diseases. She's very effectively collaborated with physicians and clinical geneticists, and she's very interested in reaching out and identifying new collaborations and exploring the EHR and BioMe to identify undiagnosed genetic diseases. Rondo is new to the IPM from last spring. He's an assistant professor in genetics and genomic science as well. His interest is coronary artery disease. He published some of the seminal papers on Mendelian randomization. So instead of these biomarkers as being correlated with disease, he developed methodologies whereby you can take these germline variants and infer causality through Mendelian randomizations. He's interested in natural selection, interested in cardiovascular disease, physiologists, biologists, and again, very eager to reach out to clinicians interested in cardiovascular disease. Girish McCartney is in the Department of Medicine, Division of Nephrology, has expertise in clinical informatics, very interested in the questions of electronic phenotyping. As a nephrologist, he's interested in looking at serum and urine biomarkers, and he's interested in leveraging the biobanks, which I think is a real potential value of biobanks, especially if we can expand them for conducting efficient clinical trials. We're too slow in enrolling people to clinical trials, and this is a potential new avenue. So he's interested in electronic phenotyping, biologists, and population health interests for this. So in terms of bridging silos, medical school to clinical care. So we've talked about how you can conflate the IPM structure with GWAS, and a lot of the high-profile papers involve that. That's been the tangible benefit thus far, but I think the real benefit is moving forward, and we can leverage the massive Mount Sinai health system through the merger in 2013. This provides an enormous capacity, unprecedented, to combine translational research with clinical care. And getting back to Warren Buffett, a great investment opportunity occurs when a marvelous business encounters a one-time huge but solvable problem. So integrating genetics, personalized medicine with clinical care is solvable, and it's something that we're very interested in doing moving forward. So one of the most direct places where this can be applied is for pharmacogenetics, and again, there's components of it for both discovery as well as implementation. And so with that, I'll hand it over to Stuart Scott.
Great. Good morning, everybody. So yes, I'm just going to introduce a little bit of some of the pharmacogenetics work that we've done in the Department of Genetics as well as the IPM. So you've heard about pharmacogenetics for a while, probably. It's been around since the 60s. And as the technology for sequencing came on board in the 2000s, we started to see the term pharmacogenomics in the literature. And I like to kind of compare this to also looking for clinical pharmacogenetics citations, because you can see there's a lag between the discovery and implementation, which is really what we kind of want to bridge here with IPM work, is like going from translational discoveries to something that we can hopefully implement clinically. So this is kind of the overall cartoon that a lot of the pharmacogenetics lectures will give you. I don't know if I don't have a Warren Buffett quote for this picture, but I should have looked for one beforehand. But the idea is that we want to find populations of patients who are going to have higher risk for either excessive toxicity that could maybe be enhanced by a dose reduction, or those who could be higher risk for a failed treatment and could be amenable to another medication. So there was a really good review on this kind of work by Mary Relling in the Nature journal Nature last winter, looking at the number of FDA-approved medications on the left that have actionable pharmacogenetic genes mentioned in their label, about 7% of them, which equates to about 84 current medications. But if you look on the right, it kind of shows you how important this is, because this is the number of prescriptions of these medications, suggesting that that number now goes from 7% to 18%, suggesting that these are not rare medications. These are medications that are commonly prescribed. So I want to just give you an example of some discovery work that we do. Judy did a really great job of talking about genome-wide association studies. Another strategy that we've implemented is sequencing exome or sequencing extreme phenotypes, as shown here in the the top right, where we look for patients that have extreme phenotypes, either like some kind of laboratory finding, and subject them to exome sequencing with the hypothesis that some of these variants are going to be clustering in some of these sides of the distribution. The phenotype that we've done this with is a collaboration with my friend and colleague John Sebastian Hulot in cardiology. He's an expert in clopidogrel pharmacogenetics, and our phenotype is ex vivo platelet aggregation. So this is a cohort that he has from France that has already been published, patients with acute coronary syndromes. So we look at we define our extreme phenotypes at the extremes of the distribution. We then make sure we genotype them. There's already a candidate gene that we know is implicated in drug response variability. So we make sure we rule that out by only selecting those who are wild type at the previous candidate gene. We then take the variants that we identify by sequencing from each extreme of the phenotype and rank them in order of importance statistically, and then go back to the entire cohort and genotype them across the entire cohort to see if anything withstands statistical significance. And in this case, we were fortunate to find a novel gene and novel variant that's currently under review at another publication. So translating this from discovery to implementation, I wanted to introduce briefly the Mount Sinai Genetic Testing Laboratory. As Judy mentioned, silos. This is one of the silo here at Mount Sinai that we've recently bridged with IPM. This is ran by Dr. Lisa Edelman. We're over in the ATRAN building. This is our CLIA certified genetic testing laboratory. I'm one of the 15 board certified directors. We've grown dramatically over the last few years. The volume has increased significantly with all of the Mount Sinai Health System expansions. But we do have a clinical pharmacogenetic testing panel. It's a 5-gene panel that we currently test for. These are genes that are involved in drug response variability in cardiovascular medications, as well as psychiatry medications and others. But there's such interest in this field now, we have a new next-generation sequencing diagnostic panel that we'll soon be coming out with. It has an expanded gene list shown here. And we hope to be implementing some of the HLA alleles as well, too, as these are really strong signals when it comes to rare adverse drug events. So understanding that the current clinicians may not have a strong educational background in pharmacogenetics, I just briefly wanted to mention before Aniwaa comes up and talks about some of the implementation efforts, some of our work in trying to get pharmacogenetics into the education system here at Mount Sinai. So what we do is when we give our genetics component to the first and second year medical students, we offer them pharmacogenetic testing at the very beginning of the year. We give them saliva kits and a consent form and a baseline questionnaire study. And it's an optional activity, and we basically just want to measure their interest and their perceived benefits and see if the actual option of getting their own genetic testing done and these results returned to them is of any use to them. So we get the kits back within the first few days. Then we have about 2 months to do the actual clinical panel testing. And then when I give them their lecture in around November, we also have their PGX reports sent back to them at the same time. They're de-identified. The school was very concerned about any kind of some kind of They didn't want the students to feel like they were coerced into doing this whatsoever. And so then we give them questionnaires longitudinally after the course. And so the preliminary results we presented at some genetics meetings recently, but their knowledge about pharmacogenetics has significantly increased. And the data does suggest that they have a much better understanding of the risks and benefits of pharmacogenetic testing. So we're really excited about it. We have funding to do it now for 3 years more. So with that, I will then pass it over to our implementation expert, Aniwaa.
Okay. So from what Stuart and Dr. Cho just spoke about, we decided to implement this in our clinics to find out how the providers will receive this and if they thought that this sort of information was relevant in the care of their patients or not. So our program is not looking at outcomes of using pharmacogenomics, but developing and establishing a best practice for clinical implementation of genomic medicine using pharmacogenomics as our pilot. So currently we have 2 programs in IPM. One is IPM PGX, which is locally funded, and then the other is eMERGE PGX, which is a part of the eMERGE network. So the eMERGE is a network of 10 different institutions across the nation, and we happen to be a part of it for the pharmacogenomics component. So as you can see, for IPM PGX, we enrolled 1,000 patients from our Biobank, whereas in the eMERGE cohort, we have about 663 patients who were part of the Biobank or patients that were enrolled de novo. So with the IPM, we use genotyping as compared to a sequencing platform that was used in the eMERGE program. Those are 2 of the main differences. The other difference is the fact that the providers in the IPM program are consented and they receive surveys from us every time they see a clinical decision support, whereas the providers in the eMERGE program are co-investigators. So that happened mainly in the faculty practice associates clinic, whereas the IPM was in the internal medicine associates clinic. And currently, this is open to all ambulatory clinical settings that are EPIC enabled in our health system as well. And both programs use ClipMerge, which I'll go over briefly. So we conducted a provider training, and these are 2 main things that I wanted to share this morning with you. That when we asked the providers if they knew enough about genetics and genomics to understand the test results on their own, we realized here that about more than 50% said that they didn't know enough. So we knew that we had to come up with a way to translate this, as well as when we asked them if they had enough experience with decision support and genome-guided prescribing, we realized that with decision support, more than 80% or over 90% said they knew how to use decision support. But when we asked the question about genome-guided prescribing, about 91% of the providers that we surveyed said that they had no idea how to do this. And this was a cohort of about 200 providers. So we thought that there has to be a way to do this. So we conduct training. This is about an hour training, but the training itself takes about 30 minutes. But beforehand, the providers go through a pre-training questionnaire and a post-training questionnaire, as well as in each CDS that the providers see, we have embedded more information and links to reference materials so that they can continue this education as well. And lastly, we survey the providers. It's a quick, less than 2-minute survey, 5 questions about whether they found the decision support useful, whether they used it, why or why not, and will they use it in the future again. So briefly, I want to go over ClipMerge. So ClipMerge stands for Clinical Implementation of Personalized Medicine through Electronic Health Records and Genomics. So ClipMerge is an advanced data management platform. So we use this as our clinical decision support engine. So this is exactly how it works. So we received the genetic information from the Mount Sinai Genetic Testing Laboratory, and this is stored in the ClipMerge database. So every time a provider opens an EPIC encounter for any of the PGX patients, we received a longitudinal clinical data, including the medications that the provider is prescribing at that point in time. So ClipMerge performs a risk assessment of the patient's genotypes and the medications that they are being prescribed. And if the patient happens to have a high-risk genotype, that would cause them to have either a side effect or inefficacy, ClipMerge sends information directly back into EPIC. This all happens in real time at the point of care. There is no lag in the workflow at all. So I'll show you some examples of this. So this is an example of a simvastatin alert in EPIC. And currently we have 5 medications that we have clinical decision support for. So they are namely clopidogrel, simvastatin, warfarin, codeine, and tramadol. So this is just an example. So in this case, the patient has a genotype, which is the CC genotype. And we explain, because we understand that just saying CC genotype does not mean a lot to a provider. So we explain with a clinical relevance of this genotype as it pertains to the medication that the patient is being prescribed. So in this instance, we tell them that this genotype confers high myopathy risk with simvastatin, particularly with doses greater than 20 milligrams. And the recommendation is always in this box, and this is the format that we use for all the genome-guided prescribing aids. So here, the recommendation is to consider prescribing a lower dose or switch to an alternative statin. And there are many links in here that I want to share with you. So you can click here as a provider for further info, and it opens a pop-up with a one-page PDF document that simply reviews the need-to-know information concerning this. If you want even further information, there are links embedded in this PDF that takes you directly to the guidelines that we used, which is the Clinical Pharmacogenetics Implementation Consortium guidelines. And there are other resources that we also use. So they are all linked in this PDF for you. So you don't need to get out of EPIC to see this. It pops up right there and then. Also, if you want to preview the SMART set, so the SMART set is a list of alternative therapies that we are suggesting that the providers can use. So if you want to preview the SMART set before accepting the recommendation, it opens up another window in EPIC as well, and it lists all the alternative statins that are available as well. And once you accept the recommendation, it opens up a window in EPIC directly in the order entry, and it copies the BPA, or the best practice advisory, so that you don't necessarily have to remember everything you saw, but we just give you another image of the BPA, and all the alternative medications are also listed, so you can easily go into any one of these and order that medication without having to go back and start the order entry process again. So every time a patient also, or provider receives a BPA, the BPA text or the language of the BPA is also reported as a result in the patient's EHR. So in this case, this patient's EHR is updated with this information. So this is also another source of reference for the providers if you want to go back and see what the patient had. And we've seen this being used. Some providers actually copy and paste this into their notes and explain and use that as their reason for switching therapy from one to the other. So we've seen this being useful as well. So this is just our acknowledgements, and we just want to thank everyone because it's been a huge team effort, and we couldn't have done it just in IPM. As you can see, there are so many other people that helped us throughout the way. Thank you.
We're going to do a panel discussion, so we'll invite our speakers to the front. And we'll open it up for questions.
Okay, yes, go ahead.
Yeah, thanks very much.
there may be an oversight in our research. I don't get the feeling that we are taking advantage of looking at the genetics of older patients, thereby to help young patients through what we learn in older patients, and of course to help older patients. Am I correct that there is very little attention to older patients in these studies?
With older patients, you said?
Well, let's call old 65.
So with the PGX program, we actually used that age group. Honestly, we picked patients that were over. So for both programs were different. One was 50 years old and above, and the other, I believe, was 65 and above. That patients that were currently on the medication. So the other difference is that the IPM group were patients who are already taking those therapies, whereas the eMERGE group were those that we predicted to be on that therapy within the next year or so. So yeah, there was an age cutoff limit, about 50.
Well, just one more moment. People over the age of 60 represent at least 50% of all of the time and money we spend in healthcare. So to not be paying that kind of attention and to hope we could learn about younger people through studying older people as well astounds me.
No, you're absolutely right. And so the Biobank is reflective of the touches to the health system. So it's peaking in the 50s and the 60s. So there's a good representation in BioMe of folks over 65, although it's actually disproportionately a little bit lower than what you're describing. But yes, I agree, it's a major issue, and there are important functional differences. You know, the immune system, clearly huge immune differences, drug side effects, so absolutely.
Well, why does that happen? Just to explain it to me, why do you think you ended up with this disproportion?
So BioMe does have, I don't have off the top of my head the fraction that are 65 and above, but BioMe does have a good representation of 65 and above.
Yes.
In my experience, the glue that has held all the different specialties together has been the focus on a particular patient. That's when we pick up the phone and we collaborate, and then, for example, in the Carcinoid and Neuroendocrine Tumor Center, which meets monthly, the focus really that pulls us together is the patient. And I'm just wondering, in your work, what is the glue that pulls everyone together? Is it a protocol, or where's the focus really?
It's a great question, and it gets back to that silo thing. What are your habits? Who are you sitting next to? Who do you have regular meetings with? And again, the financial thing kind of works for you and against you. So like, one of the best ways to get to know people really well is to write a grant together. So if you're going for shared funding together, that works extremely well. And so it really is just advertising and evangelizing, if you will, kind of what the assets are within an institution. Because and I'm as guilty of this all the time, is I didn't realize that the expert in X, Y, and Z is like one floor below me. And so it really is trying to become familiar, and this again is what we're interested here, is making people familiar with computer IT and the biobank of like what are the resources and assets right here at Sinai.
If I could add just a little bit. In the implementation work that Aniwaa described, a lot of our most important feedback came from the practicing physicians. So the people who are looking at these alerts and saying, no, that doesn't work for me, I need to know, I need it more brief, I need it more actionable, I need more explanation. So it had a tremendous amount of very valuable feedback from the people who are seeing the patients.
Yes.
I'm just wondering, for the Department of Medicine, is each division have a representative that interacts with your DOM team? And also for the whole system, the institute, have you included all the members of the merger of different hospitals, or is it Mount Sinai campus?
So that's a great question, and I kind of two parts to it, and again, if other people have thoughts. So just as a very specific example, so I showed Girish McCartney, who basically is a nephrology fellow, And so the vision I have is that we will start bringing in, again, the level of interest, the level of training that's most appropriate. I'm thinking fellowship is the way to do it, is you identify a fellow in the subspecialties who can kind of be an IPM contact. And so in a very short period, of course I'm a gastroenterologist, so we invited Ryan Angaro. So Ryan was turning in a grant that he just turned in last week, and he got help from Girish McCartney. So even though they normally would never talk to each other, the fact that they have such a shared experience, same internal medicine residency, medical subspecialty, they come from the same background, so they immediately were able to connect, plus Girish is a great guy. And so they were immediately able to work together very productively. And then in terms of the system-wide issue, great point, again, we're applying for this PMI grant, and one of the big, for numbers, you're supposed to recruit 35,000 people a year. We can only do this by including the entire system, and so I've had conversation with Bert Dreyer. If we got the grant, maybe we'll involve, I'll join their monthly meetings for the system-wide integration. Very important.
I have a question maybe for Stuart. With respect to pharmacogenomics, there seems to be variability between specialties. So for instance, thinking of TPMT mutation that the gastroenterologists check in everyone, rheumatologists never check this before azathioprine use. So I'm just wondering, do you have any thoughts on why there's this variation? How can we address that going forward throughout the system?
Sure. So one of the things that our group does is we're always evaluating the level of evidence behind what's probably the most useful for implementation, because there's a lot of signals out there that doesn't really pass the bar in terms of whether it's worthy of having some clinical utility, because defining clinical utility is really hard in pharmacogenetics too, because people always wanted to see a randomized controlled trial to prove that it's worthwhile to implement.
And cost effective. And
cost effectiveness, exactly. So we have a group that kind of meets regularly to evaluate what would be useful to implement. And so the TPMT is a good example too, because I know that there's an enzyme test as well for it too that I think a lot of people prefer to the genetic test.
Yeah, we'd love to hear more from you guys about that, whether we should do it.
Sure. Yeah. But we're always looking for feedback too on, because we don't have the bandwidth, I think, to be completely on top of all of the different subspecialties and things like that. And so we're welcome, and we welcome to get feedback on what you guys think would be important. Yeah.
So the TPMT genotype is complex, and so you should be measuring enzyme activities for sure. And so that's why people don't order the genotypes, because the TPMT genotypes are not as predictive as the distribution of TPMT enzyme activity. Having said that, but that TPMT activity is reflective of the risk for leukopenia. There's another side effect that is idiosyncratic, which is pancreatitis, which typically occurs within 6 weeks of starting azathioprine. And so there is we've talked to Stuart about this, there's an MHC class 2 variant, which is relatively common in European ancestry individuals. I'm forgetting the numbers now. But it quadruples your risk of getting this autoimmune pancreatitis, which has a high baseline risk. So again, I don't have the numbers. It's like 4% of all individuals started on azathioprine have pancreatitis. If you carry the at-risk genotype, your risk of developing pancreatitis is fourfold. We're having a raging debate in IBD as to whether or not that changes management and whether you should go to methotrexate or not. So enzymes better for blood. The MHC class 2 is probably valuable for pancreatitis.
I can tell you that less than 10% of rheumatologists are checking even the enzyme levels.
Well, so like when I was a beginning IBDologist at the University of Chicago, one of the first people that I ever put on azathioprine was a homozygote for the TPMT genotype, and thank goodness he was compliant with his blood test because his neutropenia came back immediately. So I don't know.
We need education.
Why not do it?
Great. Dr. Cole.
I was wondering if you could give us more details about how you actually use the information for warfarin and clopidogrel, because these are rather complex stories, and in fact, they're stories in which a Bayesian approach to the prior probability before the genetic testing really has a major impact on the utility of the testing. So are you incorporating what you know about the patient before doing the test into recommending what screen pops up for them?
Sure. So Great to see you here, Dr. Guller. So yeah, for clopidogrel, it's complex, as you know, and so we make sure that we don't tell you what to do. We give you the options of what the practice guidelines suggest are things to consider, because we're not cardiologists, so we don't want to be the ones to be giving a direct message that way, of course. So there's the information for you to decide whether this extra piece of information it provides you enough evidence to change your mind. So for the warfarin example, though, it is much more complex, and Aniwaa, we didn't have time to present it, but Aniwaa has done a really great job incorporating some of the known clinical factors, not just the genetics, your ethnic background, your height and weight, other medications that you're on, for those multi-dimensional dose prediction algorithms. And so that pulls that information out of Epic automatically.
Because, as you know, even with some of the CPY 2C19 variants that lead to less metabolism, a significant percentage of those patients actually have a good antiplatelet response to clopidogrel.
Yes.
And the question is it worthwhile recommending moving them to a drug that potentially has more bleeding risk and is certainly a lot more expensive if that's the case? So I just wonder how people are actually responding to the...
So that's been the challenge with 2C19, is it has a low positive predictive value. But I think the negative predictive positive value is high. The negative predictive value is high. So if you don't have the variance, there's a better chance. But it is a highly debated field.
I'm not even sure. I'm not even sure that's true. A significant fraction of the non-responders are people whose genotype is within the good categories of genotypes. So I just think it's really complex, and you know, in your case, you're doing all of the genotyping up front, right? So you're not in a situation where somebody has to make a decision about whether it's worth it in terms of the cost of doing it, and also the delay in terms of instituting therapy. So I think it's great to be doing this because we have to get more information, but I think the refinements are going to take a lot of very, very hard work and additional study.
Yeah. And we tried to implement 2C19 directly through the catheterization lab here, but you're right, the turnaround time of the testing was inadequate. They needed the results so quickly, such a chaotic testing environment. That the preemptive strategy seems to be more effective.
But then somebody has to pay for that up front.
Yes.
On a population level, which is sort of a population decision about cost effectiveness. And at least in these two areas, I think while the data are compelling in terms of associations, I think the issues of their clinical utility and cost effectiveness outside of the populations with prior probabilities of high risk, using that in a sort of functional way, I think is going to be quite difficult to sustain.
And also, in regards to clopidogrel, CMS does cover clopidogrel testing, 2C19 testing for clopidogrel if the patient is undergoing PCI. So so that example is not a universal one. The problem there is it's an indication-specific example. So if the patient has not undergone PCI, there's no recommendation for you to switch therapy, even if they have loss of function. And that's one of the main reasons why I still believe that this is a complex one, that we don't fully understand the whole underlinings of it.
Not to get into too many details, but I think that was a political decision in and of itself. And it's driven by the CDC recommendations, which focus on the genetic information and completely ignore functional testing. Even though the data with functional testing, I would argue, were as compelling or more compelling than the genetic data associations. So I think we really need to dig in and use the genetics in the greatest context of other information that we have.
I
Great.
agree.
Any further questions? If not, let's thank our panel for a fantastic